I'm here to talk to you about the basics of nucleic acids. We're going to start by talking about the molecule that we spent all the last couple of class periods learning about. That is DNA, deoxyribonucleic acid. Deoxyribonucleic acid, there's supposed to be a space right here. You're going to have to pretend like I have better handwriting. Basically, DNA, we talked about, was discovered a long time ago, but the structure of it was just finally figured out in 1953 by Watson and Crick after going around and talking to all of those other scientists that we learned about in class. So the structure then, what is the structure that they learned all this much about? We talked about how it has, it's a polymer. DNA is a polymer, which means it's a large molecule made of many small parts called monomers. And so exactly what is the monomer of DNA? The monomer is called a nucleotide. Nucleotide. Nucleotide has three main parts: the phosphate, the sugar, and the nitrogen base. This is a very basic diagram. It's not super detailed, it's not even super accurate, but it's pretty darn close. So basically there's a couple things that are really important that we need to impress upon you. The first one is that this little guy here with a number 1 on it, that is a phosphate. We all know what a phosphate looks like because we've studied our functional groups. It's the same phosphate that we discussed many, many times over again. The second part is a sugar, and specifically in DNA, that sugar is deoxyribose. Later on, we'll talk about in RNA how it's just ribose, but for now, we're going to talk about DNA, so we're going to use deoxyribose. And the third part is a nitrogen base. We learned about Chargaff and Chargaff's ratios. He was looking at the nitrogen bases, adenine, guanine, cytosine, and thymine. And eventually, we do need to know all the different names of them, so go ahead and write those down. Cytosine and thymine. Those are the 4 nitrogen bases found in DNA. There is another nitrogen base called uracil that's found only in RNA. And so in RNA, which we'll talk about again later, thymine is no longer found, but we find uracil instead. So we'll get to him a little bit later, but for now we're talking about DNA. So like I said, we'll talk about A, C, G, and T, the 4 nitrogen bases found in DNA. And what exactly is a nitrogen base? So let's look at that for just a second. These are the nitrogen bases, and if you remember on the video we watched, we saw that they looked like ring structures stuck together. I have a little secret trick that helps us remember which ones are which. We have purines and we have pyrimidines. And the purines are the ones with the double ring structure, and the pyrimidines are the ones with the single ring. So here's how I choose to remember it. Girls are the kind of girls who are pure, right? We're supposed to be pure. And that means that we wear a wedding ring when we get married. Well, we wear 2 wedding rings. We have an engagement ring, and we have the wedding band. So we have 2 rings. Guys, on the other hand, they don't get an engagement ring. They just get a ring. Just get 1 ring. Guys, the boys with a Y in their name. See that Y? There's another one right here, the Y. Guys also has a Y. So if you say boys, you say guys, it doesn't matter. There's a Y in the name. There's also a Y in cytosine and a Y in thymine. And purple is my favorite color. Just so happens to be the color of a great university, TCU. Thymine, cytosine, uracil. All 3 of those are pyrimidines. And of course the other 2 by process of elimination. A and G, it's all good. So that's how I keep my purines and pyrimidines straight. That's how I remember the structure of the purines having 2 rings and then the pyrimidines only having 1 ring. Hopefully that'll help you out a little bit. So going back over here, the other interesting thing that we find out is that A always binds with T, always together. And G binds with C. Good combination. So, what does that mean? What does it mean they bind together? Well, when DNA forms a polymer, like I said, it's a polymer of large a large molecule made up of many small pieces, the first step is to make sure that these nucleotides can stick together. And so by doing that, they form a bond right here between the phosphate of one nucleotide and the sugar of another. And that bond is called a phosphodiester bond. And I'm sure you can think of what the name of the process is that causes that bond to occur. It's the same process that we find in every other large molecule polymerization. It's called dehydration synthesis. You've heard of it before. So as we're building a polymer, we're putting phosphodiester bonds between the individual nucleotides. Well, if you recall, DNA is double-stranded. So we've got to have one strand on the left and the other strand on the right. So the nitrogen bases are held together in the middle with a hydrogen bond right here. Let me draw the rest of my nucleotide. It's a little tricky to draw it upside down. Also, if you're watching this video and you're a biochemist and you realize that I'm not drawing my phosphate in the right place, you're just going to have to forgive me. I'm teaching freshmen, and I'm going to get to the 5' and 3' carbons in a little while, but for now we're going to have to draw it the best we can. So if you don't know what I'm talking about, don't worry about it. Alright, so here's my phosphodiester bond again. So you can see that this particular strand is facing this way, and this strand is facing this way. And this is the thing that Watson and Crick were having such a hard time with figuring out. They were still trying to put all the pieces of the puzzle together, and when they realized one of them was running in the opposite direction, they figured it out. So again, here's my adenine binding to a thymine, because A always binds with T. Here's a guanine binding to a cytosine. It's G binding with C. And this whole entire structure here is the polymer of DNA. Now, if you recall, one of the things we learned when we were talking about Rosalind Franklin's X-ray crystallography photograph or image is that we learned that there was a specific pattern of these little lines that we saw. And I'm not doing it justice, I'm trying my best here, but we kind of saw this idea. One of the things that Watson and Crick did is measure the distance across this entire molecule, and they figured out that it was 20 angstroms across, and it was consistent all the way down the entire molecule. And that gave them a huge clue, because they realized if they try to put two purines, if this is a hexagon, forgive me, if that's a hexagon and this is a pentagon, they tried to put two purines together, the width was one thing. But if they tried to put two pyrimidines together with a hydrogen bond in the middle, it was a different width. So what we had to realize is that we had to put one big molecule and one small molecule, and then the other direction, one big one and one small one. So this would keep everything the same width apart. That's why this picture that Rosalind Franklin did was so important. If I could draw that even better, you'd see what I mean. So there we go. They're the same width. That was a very important fact that they needed to figure out the structure of DNA. So that is why adenine and thymine bind together, guanine and cytosine bind together. That, coupled with the evidence from Chargaff's famous ratios, told us that there was always an equal amount of adenine and thymine and equal amounts of guanine and cytosine in any given organism. That was another very important piece of information that we learned. It's supposed to be a star. I'm doing real good today. That we learned when we learned about the structure of DNA being discovered. So basically, you know that DNA is a polymer that is made up of nucleotides, which are called the monomers of DNA. We learned that they are held together with a phosphodiester bond and also a hydrogen bond between the two nitrogen bases. Just a kind of overview of the difference between DNA, a nucleic acid, and RNA, another nucleic acid. DNA is double-stranded. It's got two complementary strands running next to each other. RNA, on the other hand, is single-stranded. We'll talk more about RNA later. It's very similar to DNA, but again, a little bit different. The other thing about DNA is that it contains thymine as a nucleic acid. I'm sorry, it's a nitrogen base. It's been a long day. Thymine, and then RNA contains uracil. And then the last thing is, in eukaryotic cells, there's a nuclear membrane. The DNA never leaves the nucleus. In eukaryotic cells, that would be nucleus. Cells of animals, plants, fungus, and protists. Anything with a nucleus. If the cell has a nucleus, DNA doesn't leave. RNA, on the other hand, it does leave the nucleus in eukaryotic cells. There are some other really important differences, but for now, these are the ones we're going to focus on just for this basic introduction. I hope you've enjoyed my video. You can watch it again and again and laugh at my goofy molecules and every other thing I said that was silly. Until next time, I will talk to you later and have a great weekend.